title of the talk, Computational Discovery of Significantly Mutated Genes and Pathways in Cancer. Let's try to have lots of questions from the younger half of the population. Okay? All right, Fabio, whenever you want. I mean, we can still...
I guess I can start slowly,
okay.
Just, you know, to get it going. So I guess, as Francesca said, it would be nice to have a really interactive session. I guess this is also a tutorial more than a research talk. So I'm not going to speak too much about my own work. Of course, it will appear, since it is a topic that I know fairly well. But I will mention many other methods and softwares that are related to the problem that I'm going to describe. So feel free to ask questions, since I will not get into too many details for each of the methods. And maybe even without effort, I will give a presentation that will you know, make such presentation that a lot of questions will come from you. So maybe it may be confusing enough to raise a lot of questions. Okay. I have, for full disclosure, I have to say that for this topic, actually, I coordinated with my postdoctoral mentor, Ben Raphael, that spoke yesterday. I've been told that he said that I picked the nice topic, while he said he was left with a topic that was not as nice. I'm not sure if it is true or not. Maybe later on you guys can vote on which one you prefer. So at the end.
A great show, though, Fabio. You have to be careful now, because if you don't set up a competition between Ben's talk,
no. I'm not going. So let me clarify. First clarification for the talk: it's not a competition about the talks. It's just about the topic. Okay. So I'm not even trying to compete with Ben talk. It's just about the topic. Keep that in mind when you vote after the session. Okay. Good. So let's start. And what I'm going to talk about in this talk are mostly mutations, that are changes in the DNA, and in particular changes in the DNA that appear during the lifetime of an individual. And these are called somatic mutations. So these are changes that appear after an individual is born, and mainly during the replication of DNA, during the division of cells. And these mutations, these alterations, can appear for a variety of reasons. That can be even just error in the process of DNA replications, or other reasons as well. So the mutations that I'm going to consider and I'm going to describe in general could be at different scale in the genome. They could be single nucleotide variants, change of just one base in the DNA. They could be larger scale mutations and alterations, like deletions of big portions of a chromosome, duplications or inversions or translocations. In general, I'm going to speak about mutations, meaning any of these types. Of course, in some cases, it will be mostly related to only one of them. If you are in doubt to which one I'm referring to, please ask. Okay, so why are we interested in somatic mutations? Probably you have already heard this during Ben's talk. And the reason is that the current theory for how cancer arises is that starting from a normal cell, during the lifetime of this cell, it accumulates some mutations that are represented here by these flashes. So these black flashes are mutations in DNA of the cell that do not change the way the cell functions. So they are random passenger mutations, while some mutations actually confer a selective advantage to the cell and to the cells that have the mutation, and are represented by these red flashes that are driver mutations. So once a cell is hit by a driver mutation, that it generates a population of cells containing all the mutations that were present in the cell that was hit by the driver mutation. And now we have a population of cells that will accumulate some random mutations, passenger mutations, some driver mutations that instead confer a selective advantage to some of the cell. And this process goes on until at the end we have what's a tumor, that is a population of cells that is now different from the original founder cell, even if its DNA is related to the one of the initial founder cell. Okay, so thanks to the advances in DNA sequencing technologies, now we can take the tumor mass, put it in the cancer genome, and through a process that I'm gonna essentially skip in all the details, we can obtain the mutations that we see in the tumor mass of a patient, okay? So by sequencing the tumor mass of a patient and actually comparing it with the normal tissue or with blood for that patient, we can see what are the mutations in the patients, or in case we are doing whole exome sequencing, we will see what are the mutations in the genes of the patient. Of course, by sequencing, we don't see which mutations are driver and which one are passenger. We only see mutations. And just to give an idea of the numbers that have been, the statistics that have been collected recently. So in the first complete cancer genome sequence has been obtained in 2008. And from recent cancer studies, like typical numbers are more or less the ones that I've shown here, that are, there are maybe around 10 driver mutations or something in that ballpark. So let's say few driver mutations in one tumor, while there may be hundreds to even tens of thousands of mutations in total in one tumor, okay? And of course, one of the challenges when, yes?
Quick question, how do you tell the difference between a driver mutation and passenger mutation?
Great question, and in particular, this is exactly the challenge that I wanna talk about. So that's the perfect question, yeah.
You keep going. I also wanted to try and see if we can clarify something. So, in the order of 10 driver mutations,
Yeah.
in the order of hundreds to tens of thousands of passenger mutations,
Yeah.
would that be in some way comparable to the rate of somatic changes in non-tumor cells, or is it heightened greatly?
So this is the mutation rate in a tumor is definitely higher than in somatic cells. So it's like, I think at least 2 to 3 order of magnitude larger than what you observe in normal cells. Okay. Yep. So getting back to the question, we don't know which mutations are drivers, which ones are passengers, just by looking at them. Actually, this is a, let's say, biological property. So you will need to see biological difference in a cell with a mutation compared to the cell without the mutation. And what I'm going to talk about are methods that try to prioritize mutations so that they can be validated in the lab. Okay, so the same question, the one of finding driver mutations, can also be posed as finding driver genes, or the genes that are hit by driver mutations. Okay. And since the cost of sequencing a cancer genome has been down a lot recently, so I guess I've seen this slide, this graph was present also in the previous talk, and I was actually surprised, I have to say, because in the recent talks in computational biology now, this is not even something that you need to show. So now I'm going back to showing it because every time I can say I'm the first one presenting it into this workshop or conference, but this time I wasn't the first one. It is not even the first of today, I have to say. But anyway, cost of sequencing has been down by a lot. So what we can do, instead of obtaining mutations for only one patient, we can obtain mutations for, let's say, many patients, and try to leverage on all these data sets, or on the large data sets that we can get, to identify patterns that will correspond to driver mutations. So to give you an idea of actual numbers, so there are at least 2 large studies that have collected and made available publicly mutations in many cancer patients, like the Cancer Genome Atlas and the International Cancer Genome Consortium. So in the last release from the International Cancer Genome Consortium, ICGC, it's from June 8th, and there are mutations measured in more than 12 tumors, so more than 12 patients, and of at least 21 cancer types. So you can go, to the website and download this data and start analyzing it. Okay, so what I'm going to talk about in the rest of the talk are different methods to identify driver genes, genes containing driver mutations, talking about different classes of methods. The first class is the class that look at single genes, so genes individually. These are single gene methods. And then I'm going to talk about methods that look at combinations of genes in trying to go beyond some of the issues and limitations of single gene methods. And for the gene combination, I'm going to talk about 3 classes, the ones that use predefined combinations of genes, the ones that use networks, and the ones that use combinatorial properties of mutations, in particular exclusivity. So let's start with single gene methods. So single gene methods are methods that look for recurrently mutated genes. And are based on the idea and the fact that somatic mutations in different patients are independent events. So contrary to the variants that you see in the population that are related into different individuals, the mutations, the somatic mutations that you see in two different cancer patients, they are independent events. That means they are related by the fact that they arise from the same process that leads to cancer, or they are independent occurrences from that process, okay? And so since somatic mutations are independent events, what we can do to identify driver mutations is to look for recurrently mutated genes and hope that in this way we'll find functional driver mutations. So what does it mean to look for recurrently mutated genes? In first approximation, we can think that if we have the mutations measured in many patients, let's say in the genes, we can transform this data into a mutation matrix that has now patients on the rows and genes on the columns. And we have a 0 in this binary mutation matrix if the patient has no mutation in the gene, and a 1 instead if the patient has a mutation in the gene. And now we look at one column of this matrix, and we try to understand if the number of mutations that we see is high. If the number of mutations that we see is high, then we say that that gene contains driver mutations. Okay? So this is the idea. Yes?
So in these calculations of number of mutations, do you also adjust for the length of the genes?
Yeah. So we're going to get there in a couple of slides. You don't really look just for the frequency of mutation of the gene, number of mutated patients. You have to take into account a baseline mutation rate and to take into account for other characteristics of the genes, like the gene length and other ones as well. Okay. Okay. So as we just said, we don't really look just at the frequency of mutation of the genes. What we are going to do is just perform a statistical test on each gene and then correct for the fact that we are testing many genes. So I'm going to give you one slide on statistical hypothesis testing. It's really risky, I guess, since there are many statisticians in the room. So in particular, Francesca is sitting in the first row. So take it as an approximation of introduction to statistical hypothesis testing. And so in statistical hypothesis testing, what we have is a null hypothesis, H0, and we have some data that we have observed, D. And our null hypothesis in general corresponds to the fact that the phenomenon is not interesting. Okay? This is the way to set it up. And then given the data, we have a test statistic that we can think of as a function of the data, just as an observation. And what we want to compute is the probability of obtaining a test statistic at least as extreme as the one that we see for our data set, assuming that the null hypothesis is true. And this is called the p-value for our hypothesis. Okay. Now, if the p-value is small, or small usually means below a threshold alpha that can be fixed in different ways, traditionally 0.05, then you can choose your own threshold. It should be picked before you look at the p-values. So if the p-value is small enough, you reject the null hypothesis. So you say that your observation is significant, essentially. And doing this guarantees that the probability of a false discovery, so the probability of rejecting the null hypothesis when it is true, will be less or equal than alpha. Okay. Okay. So just to give a concrete example, I'm a computer scientist, so when we give examples with probabilities, it's always about coins. Okay, so in this very complicated example, let's say that our null hypothesis is that the coin is fair. And now my data consists of the fact that I have observed, I've flipped the coin 100 times, and I've seen it coming up with head 75 times. Okay, so now my function f of d is, for example, the deviation from the expected number of heads expected under the null hypothesis. So in this case, it's 25, because I have 75 instead of 50. Okay, so now I can compute the p-value, that is the probability of obtaining a deviation from the expectation that is greater or equal than 25 when the coin is fair. This probability will be, if you do the computation, is less than 10 to the minus 6. It's a very small p-value, So what I will say is that this coin is biased. So I reject the null hypothesis. Okay. We're almost done with our statistical hypothesis testing 001. It's not even 101, I guess. And the only other concept I want to introduce is the one of multiple hypothesis correction. So here we have considered only one hypothesis, H0. But in general, we may be testing multiple hypotheses. So in our example, we may have not only one coin, but a number of coins. Or if we go back to our example of single genes, so instead of looking at only one gene, we're looking at a number of genes. Okay? So what changes when you have multiple hypotheses that you are testing is that you have to correct for the number of hypotheses that you are considering. Since if you have m hypotheses that you are testing, and you use the exact same algorithm used above to reject null hypotheses, then the expected number of false discoveries will be alpha times m. Okay, so for example, if you're testing 1,000 hypotheses and your alpha is 0.1, then you have 100 false discoveries in expectation. So if you want to control the number of false discoveries, you have to take into account the number of hypotheses that you're testing. There are different ways to do it. Just the simplest way to perform what's called Bonferroni correction, or for computer scientists to do a union bound. That means you reject the null hypothesis if the p-value is less or equal than alpha divided by m. And this guarantees that the probability of having even one false discovery will be less than alpha. Okay? And there are other corrections that you can do that will bound the family-wise error rate, that is this probability, or the false discovery rate, and there are more complicated schemes. But one thing to keep in mind is that you need to correct for the number of hypotheses that you are testing somehow. Okay, so for single gene tests, what we can do then is the following. Let's say we have m patients and n genes. I can look at one gene, a specific column of this matrix, and now I observe in how many patients it is mutated. Let's say there are R patients in which the gene is mutated. And let's say that I know the passenger mutation probability for gene G, that is Q of G. This is the probability of observing a mutation in the gene when the gene is hit only by passenger mutations. Okay? So if I know Q of G, I know the number of patients, and I know the number of hypotheses I have. For example, the quantity I want to compute, the p-value, is the probability that x, that is down the number of m-independent coin flips when the probability of having had is Q of G, is greater or equal than R. Okay. If this probability is less or equal than alpha divided by n, I can reject the null hypothesis. Now, this x is a random variable that follows a binomial distribution, and so I actually know how to compute such probability. And this is a first simple algorithm, a first simple scheme to identify significantly mutated genes, recurrently mutated genes. It looks very simple, but it is actually what's used in all large cancer studies with some minor, not really minor, with some important changes that I'm going to mention in the next slide. But now, given the genes that I'm looking at, I can compute the probability according to my binomial distribution with the right parameters. And at the end, what I get is a small number of genes that are mutated with quite high frequencies in the cancer samples that I had. So for example, let's say that this is a figure from a study from the Cancer Genome Atlas in 2015. And using the approach that we described, they identified 9 genes that are significantly mutated. And this is the frequency of mutation, so the fraction of patients, of the 127 patients in which the gene is mutated. Okay? So you see that this approach identifies genes with quite high frequency. Okay? Yes?
How do you calculate the norm distribution?
Yeah, that's a great question. And that's the most important question. Okay? And we're going to give you an idea like in the next slide. Okay. So this is a basic scheme, and there are actually many different methods that use this basic scheme of using a binomial distribution, essentially. And the main difference between them is exactly what you just asked for. That is, how do you compute this passenger mutation probability, the mutation probability for a gene G? Well, computing such probability is a difficult problem because you need to estimate it from the data. And that's really where the difference between the methods stands. And so let me give you a few words about different approaches for computing this passenger mutation probability. And in particular, I want to tell you what are the biological features of a gene that influence its passenger mutation probability. Okay, so just to recapitulate, what we want to do is be able to compute the probability of observing at least R mutations, assuming that there are no driver mutations in a gene. So what we need is the number of samples or patients that we are observing, and we need this background, this passenger mutation probability, that is the probability that the gene G is mutated in a patient by chance, that is related to the background mutation rate for the basis in the gene. Okay, so the main challenge is that we need to estimate this parameter, the background mutation rate. It's a very difficult problem, and usually it's done by looking at the rate of silent mutations. So if I look at a gene, I consider silent mutations, mutations that do not change the protein obtained by that gene. I look at how many silent mutations I have, how many bases for the gene I have sequenced, and the ratio between the two gives me the rate of silent mutations for that gene. And usually that's my estimate for the background mutation rate for that gene. Okay?
So it's gene specific, but it's the same across the whole gene. It's going to be
Yes,
the same across.
exactly. It's gene specific, but it's the same for all gene. It's the same in all patients as well.
Ah.
While instead you have a different, could have a different background mutation rate in different patients as well. Okay. Yeah.
But, so this is basically, you're looking in introns, or this is only exons that you're looking
This is essentially only exons, because most of the data comes from all exome
at? So
sequencing.
you're always looking at third position, basically.
Yeah, more or less. Yeah.
But there's different number of possible silent mutations depending on the amino acid. So
Yeah, so you can correct for that as well. You can count the different number of combinations that you can have and see how many of those combinations you actually observe. And again, this is estimated from the data. The events that you observe are quite rare in general, so your estimates will not be extremely reliable. Also because there are actually many other parameters that influence this background mutation rate. For example, somebody mentioned earlier, in general, to compute the probability for one gene, you need to consider its gene length. So when you compute this probability QG for the gene depends on the gene length. You have to take that into account. It's a simple parameter to consider, but there are other ones that maybe are not intuitive, like the expression of a gene and the time to replication for a gene. So actually there are studies. This is a study from Lawrence et al. In Nature 2013 that shows that the mutation rate across the genome, this is for chromosome 14, the mutation rate is actually well correlated with the low expression for the gene and the late replication time for the portion of the genome. And so you need essentially to take these other covariates or features of your genome into account when you estimate the background mutation rate. And there are more recent papers that show that there are also other covariates, like the occupation time of proteins on the genome. So there are many parameters that you should take into account.
Certainly there are many studies that investigate these type of things for germline mutations.
Mm-hmm.
And there are lots of things that affect the local rate of mutations and also the covariation of different types of
Yep.
mutations. I guess that for somatic mutations, there are similar studies. The thing that I was asking before was, so we assume that this is going to depend on where our gene is in this landscape of mutability, let's put it like that.
Yep.
But within the length of the gene across its exons, we assume that things stay the same.
Usually you do that, yes. Even if there are recent studies that show that different parts of the gene may be mutated at different rates, and it's maybe even more complicated. Different parts of the gene may act as an oncogene or as tumor suppressor, depending on where you have the mutation. Okay. Yep. So what's the baseline and the punchline actually for what I'm saying here is that with this approach relies on your estimate of the mutation probability for a gene G. So it will never be very reliable. So with this approach, you find driver genes that are mutated in a large fraction of the patients. While instead is difficult, I would claim almost impossible to use this approach to find driver genes that are mutated in a small fraction of all the samples. Okay? So how can we find then these driver mutations mutated in a small fraction of all the samples? Well, I'm going to talk about methods that look at combinations of genes, and in looking at combination of genes, they try to find and identify mutations that have a lower frequency of mutation compared to single gene methods. Okay? So this is the goal of these gene combination methods. I guess I have 15 minutes, so now I'm going to try at least to cover some of these approaches, maybe the first two. Okay, so what's the idea? Why do we want to look at gene combinations? Well, one reason is that cancer is a so-called disease of pathways. So the cell is essentially circuitry of interacting proteins and the corresponding genes. And by interacting, a group of genes actually performs a function in the cell. And to develop a tumor, you don't really need to change only one gene. What you need to do is to alter the function. And in particular, you need to alter a certain number of functions. This may correspond, for example, to the hallmarks of cancer. Okay? So there are a number of functions that need to be altered to develop cancer. These functions are performed by groups of genes. So what we want to do really is to identify the groups of genes, the pathways, that are significantly mutated. Not just the single genes, but the groups. Okay, so how can we do that? Well, there are different methods. The first one looks at predefined combinations, predefined gene sets. So what's the standard practice, essentially, in all the cancer studies, is to assess the enrichment of mutations on known pathways or set of genes. So there are databases of set of genes that perform different functions, and you can look for enrichment of mutations into these sets. For example, what you can do is create a metagene by grouping together the mutations. So let's say you have a group of 3 genes that correspond to these 3 columns. You put them together, and now you see that this set is mutated in the first sample, the second one, and this other sample. Mutated for a group means it has at least one mutation in a sample. Okay, and now you can apply the same test that we have seen before. Okay? So the problem with this approach is that there are usually there are few highly significantly mutated genes that can skew the test. So all the sets that will contain the such highly significantly mutated genes will look significant. The ones that do not will not appear significant. Yes?
The gene sets are not overlapping?
They are overlapping, but you can consider them as independent, and then you do your Bonferroni correction that kills every correlation that you can have, kills also every significance probably that you can get. So there are also more advanced approaches, but the basic idea is essentially this. Okay? So this is one approach. There are more advanced ones. For example, you can determine a list of mutated genes and now assess whether in your set, in your predefined set, you see more genes from the list than you would expect using an hypergeometric test. This is done commonly by tools like David and gProfiler. They are used quite commonly, and this is a simplified version of what they do. They take into account the fact gene ontologies are hierarchies, so they are more complicated than this, but this is the basic idea. Okay? And then you can do even more complicated things like gene set enrichment analysis, in which you rank a list of genes according to a mutation score, for example, for each gene, and now you compute a given statistic starting from the top of the rank. And you can use other methods like the Kolmogorov-Smirnov test to identify, or let's say to compute a p-value once you use this test. Okay? These are all methods that are used in cancer studies. Again, these are based on predefined sets. Okay? So to summarize these last 3 slides, what we have done is say that we have known pathways from databases or predefined by somebody. We have our mutations, and we look at each of them one after the other. We can compute a p-value and identify the ones that are significant. This is a good approach, but of course there are problems that are, first of all, not all the pathways are known. We don't know the set of genes that perform the functions in a cell and also in cancer. These pathways actually interact. They are not separate entities that do not have interactions. And sometimes these sets are very large, while only a small part of the set may be important for cancer, and these approaches may not be able to identify the subsets of a large set that is important for cancer. Okay? So what we want to do is, instead of looking at known set of genes, we want to just identify significantly mutated set of genes without restricting to known pathways. Okay? How can we do that? Well, what we could do naively is consider all possible sets of genes and perform a statistical set on each of them, and then see what we get at the end. Would this approach work? No. Why not? Because there are many candidate sets. So even if we look at only sets with at most 6 genes, there are tons of sets, 10 to the 20, more than 10 to the 22. So computationally and statistically, it's not feasible to use such approach. So what do people do? Well, what people, and by people I also include myself, do is to use some other prior knowledge to restrict the number of candidate sets that you look at. So for example, you can use a network, a large protein-protein interaction network, as prior knowledge, or you can use knowledge on the type of patterns that you expect to see in sets to identify significant set of genes.
So you don't really use predefined groups, but you use prior knowledge to choose some groups. That
Exactly.
Okay.
Exactly, yeah. And even by using some prior knowledge to choose the groups, you need to be careful in the way you do it. For example, let's start from the network methods. So let's say we have a large protein-protein interaction network, or we can represent it as a graph, and maybe instead of the proteins, we use the genes, since we are talking about mutations in genes. And we have the mutations in the number of cancer patients that we have observed. The first naive approach to use this protein-protein interaction network as prior knowledge to find significantly mutated sets of interacting genes is, for example, to extract all connected subnetworks, maybe up to a given size, 5 or 6, and then test each of these as a candidate. Compute a p-value, correct for multiple hypotheses. So, for example, we may be able to say that these two subnetworks are significantly mutated with this approach. Okay? Essentially, it's the same as before, just the candidates are not given a priori. We extract them from a large protein-protein interaction network. There are problems with this approach as well. That is, the number of candidates, the number of subnetworks that we need to test is still way too large. So it's still like 10 to the 14 candidate subnetworks with at most 6 genes for reasonably large subnetworks. And also the topology of the network is ignored. It means that when we look at a set of genes, we consider it just as a bag of genes. We discard the interactions between the genes and also the interaction of those genes with the rest of the network. Okay. So trying to go beyond this simple first approach, people have developed new methods. So the first approach that appeared essentially is the one that is presented in the method Netbox that does the following. So we are in input, we have our binary mutation matrix, we have this graph of genes and their interactions, and it finds modules of frequently mutated genes. So what it means is that first it decides which genes are frequently mutated. It places them on the network. Now it discards the frequency of the genes. They are just frequently mutated. And tries to find dense subgraphs that contains such genes. Okay? It finds some reasonable modules in some studies, but there are problems with this approach. First of all, that it only looks at frequently mutated genes. So you have to decide a threshold for genes to be used in the network. And second, again, the topology of the subgraph is discarded, and in particular is not combined with the frequency of the genes. While both type of data gives you some signal. So the frequency of the genes gives you some indication if the gene is significant or not, and the topology of the subnetwork, again, can be used to infer some signal regarding of group of genes. So what I've done when I was a postdoc at Brown University, working with Eli Upfal and Ben Raphael, is we have developed a method called HATNet, that then became HATNet 2 in a more recent paper, in which we combine, let's say, a score for each gene that captures the frequency of mutation of the gene or some other score, it combines this score with the topology of the network using a diffusion process on a graph. I'm going to give you, not giving you the details of this, but the idea is that you use the diffusion process starting from the scores on the genes, so that at the end you identify hot regions of the subnetwork, where being hot depends both on the frequency of the genes and on the topology of the subnetwork. Okay. And also in these approaches we have a statistical test to bound the false discovery rate that is not just a naive test, since again you will not have power to discover significant subnetworks if you do it naively. So just to give you an idea of the type of results that we obtain with this approach, so we use HotNet2 to analyze more than 3,000 samples from 12 cancer types from the Cancer Genome Atlas, and in output we obtain 16 subnetworks for a total of 147 genes. This is a small figure showing the different subnetworks. You cannot really read the gene names, but some of these subnetworks contain very well-known cancer genes. Just to show you one specific subnetwork, this is a subnetwork of 7 genes that corresponds to part of the so-called cohesin complex. And so these are the genes, and this is a color coding of mutations in the different patients. So patients here are on the column, and there is a color if the gene is mutated in that patient. Different colors correspond to different cancer types. Okay? So you can see that in total, the subnetwork is mutated in 7% of all samples. Each of the genes is mutated at a much smaller frequency. Overall, the subnetwork is mutated in a significant number of patients. And here we have that the genes have a star to their left if they are not identified by single gene tests. Okay. So the main point is that with our approach, we identify these 5 genes that are candidate for being driver genes that are not identified with single gene tests. And it's not by chance that these are the genes mutated with lower frequency in the subnetwork. Okay? So we actually do find more driver genes than single gene tests. Okay. So there are many other network approaches that have been developed. I'm just going to mention one of them that is called tie-dye. That is an interesting method because it uses this diffusion process starting from mutated genes and also starting from differentially expressed genes. And now it runs the diffusion process from both in order to identify genes that are connecting the mutated genes with the differentially expressed genes in a cancer cohort. So it's an interesting application of this diffusion process on networks. Again, this is a nice approach that, however, works only on known pathways. So it has been used only for small sets of genes, predefined sets, but it gives some interesting results. Okay, so this is all for network methods. Very quick, just to give you an idea of what has been done. And now in the last 5 minutes, I'm going to talk a little bit about a different class of methods that use combinatorial patterns of mutations. And in particular, these are methods that are based on the property of exclusivity of driver mutations. So when we look at a driver pathway, so a set of interacting genes that perform some function, since driver mutations are rare, very frequently a cancer pathway has only one driver mutation in a patient. Okay? So this is an observation, and this is a figure actually taken from a paper of 2007 that shows that for the genes in this pathway, there is, in all the patients, there is essentially only one of these genes that is mutated. And this is a known cancer pathway. Okay? Another property of cancer pathways, and we call this property exclusivity of mutations. So in a patient, there should be only one driver mutation in a cancer pathway. The second property.
Because they're rare, or is there some other reason?
There may be other reasons. So there are 2 main possibilities that are considered. One is that they are rare. The second one is actually if you had 2 mutations in a driver pathway, then the cell may change too much, just to be informal. And so actually, die. Yeah. Yes, instead of becoming a tumor, it will die. So that may be the other reason. There are some known cases of synthetic lethality, synthetic lethal mutations in cancer, but in general, we don't know why we observe this property of exclusivity.
With how old is the
It also has something to do, it is correlated for sure with how old the cancer is. So the older the cancer is, the higher is the chance to have also some passenger mutation, so you would observe more, let's say, less exclusivity in a cancer pathway. Okay, so exclusivity is the first property, and the second property is what we call coverage. That means most patients have a mutation in an important cancer pathway. So cancer pathway should be mutated in many patients. So using these two properties, we developed a combinatorial approach that looks for exclusive submatrices of our mutation matrix to identify driver pathways. So I'm not going, since the time is running out, I'm not going to go through the details, but at the end, what you can do is say that you want to find a set of columns that show high exclusivity. So for most patients, there is only one column in the set that has a mutation. But of course, you can tolerate some non-exclusivity in the set. You can capture this, for example, by a weight that considers the total coverage of the patient, and it subtracts the non-exclusivity, the number of non-exclusive mutations. That gives you a weight for the set. And now you can look for approximately exclusive sets looking at high coverage submatrices, where the coverage is what we just defined. So actually we developed an algorithm called Dendrix for de novo driver exclusivity that uses a Monte Carlo Markov chain approach to find exclusive sets of genes. It has some interesting guarantees on the convergence of the Markov chain, and it has some rigorous guarantees as well. So from the theoretical point of view, it's quite interesting. Of course, we were interested also in understanding if looking for exclusive set of genes would correspond to find pathways or part of cancer pathways in real data. So we applied Dendrix on 84 samples from glioblastoma multiforme from the Cancer Genome Atlas. So this is the first set identified by Dendrix. There is also a permutation test that you can use to make sure that what you see is not just random noise. And so these are the first three genes. In red, there are the co-occurring mutations. So this is the non-exclusivity part. Then you remove this set of genes, and you obtain that the next set of genes you identify is this one. And then you remove it, and you identify this set. So what you get at the end is that if you look at the genes in these sets, actually corresponds to parts of 3 non-cancer pathways that were manually annotated in the same, in this paper by the Cancer Genome Atlas. So again, we identify part of interacting sets of genes or pathways without considering any interaction information. Okay? And so this is, if you want, a proof of concept that actually mutual exclusivity can be used to identify driver pathways. And then there has been a lot of work, subsequent work on using exclusivity to identify driver pathways. One such approach is called COMET. This is a statistical approach in which we extended the Fisher-Exact test to consider more than 2 variables. And it has an interesting combinatorial problem that it's trying to solve, but still, I think there is still a lot of work to be done in this area to find exclusive sets of genes. And when I said there are many other methods, that means there are really tons of other methods based on exclusivity. So these are some of them. I guess you can see later on the slides, exactly what are the details. Some of these sets use only exclusivity, of these approaches, use only exclusivity. Some of them use exclusivity and networks, like MIMO, for example. Some other use exclusivity to identify progression and the order of mutations in cancer, like these 2 papers. And there are more recent papers that combine also network, exclusivity, and a lot of other things. Okay, with this I'm essentially done. I just want to say that there is a lot of data available, both from the Cancer Genome Atlas and from the International Cancer Genome Consortium. I only spoke about mutations that are only part of the data that is available. There is tons of other data that should be combined and integrated properly with mutations to be able to identify driver genes and driver mutations. And this is still an open problem how to obtain something that is robust, scalable, and for which we can obtain meaningful results on real data. Okay, so let me just acknowledge the people I work with the most. For the paper I presented are Ben Rafael and Eli Upfal Brown University. Ben Raphael now is at Princeton University. Max Leiserson and Sinta Wu, that were two PhD students of Ben Raphael when he was at Brown. And then this has been funded by the NSF, University of Southern Denmark, University of Padua. So thank you, and if you have additional questions, I would be happy to answer.
Thanks for the talk. It was really interesting. I was specifically interested in the challenge that you mentioned of certain pathways not having only identifying certain parts of a pathway. And so it seems like if a transcription factor is mutated, it can potentially affect many genes in the network, even if the other genes are not mutated. So I was wondering, have you thought about using something like gene expression data to, I guess, to see whether the expression of other genes in the network have changed to like increase power to detect?
Yeah, that's a great point, and I'm working with a colleague of mine in actually formalizing this problem of identifying set of genes where you may have only one mutation in the set, but you observe, let's say, consistent gene expression changes in the set. And that's an interesting computational problem. At the moment, we don't have a good solution, so I'm not sure that we have really done it, as you're saying, but that's definitely an interesting direction for future research. Okay, while we wait for the next question, if there is any, I put some additional references. These are reviews. These are reviews so that you find all the methods. Yeah.
So actually, I had a question that had to do with this dendrit thing, this algorithm that you created to essentially partition columns of a matrix based on exclusivity, right? Other applications, other type of problems that you have applied a similar thing to?
Well, there is something that I'm working on, and actually a recent paper, or in 2016, a paper came out in find this exclusive mutation.
Fuzzily exclusive, because there
Let's
are
say
many.
fuzzily. Yeah, let's say fuzzily exclusive. Oh, it's a good name. So fuzzily exclusive set of mutations. To identify these sets that are also correlated with some functional measure for a set of patients. So, for example, drug response or something else.
Generally, that's a matrix of zeros and ones, right?
Yeah.
It's got to have tons of applications.
Yeah, so actually I'm trying to find a good data set for data mining purposes or market basket analysis where I can do that type of analysis. I think there is there are many interesting problems you can come up with, but I don't know. So in the data mining community, it has not been explored, this problem, and I have to say that I haven't tried that on those data sets. So I don't know if we will get something interesting or not, but there could be other applications.
That it's very interesting is that you can combine it with a tool to make groups, to sort of break the group in subparts that make sense in terms of its exclusivity.
Yeah, definitely. Yeah. So it's very general. And
We have another question.
So I guess my question's kind of like a two-parter. First, how often is it the case where multiple mutations will occur in a gene for a given individual? And then the second part, the follow-up of that would be like, is there any power to be gained by, instead of just considering this binary matrix, to go to a count matrix?
yeah, so that's a great question. And in most cases, for the data sets I've worked with, we observe only one mutation in a gene in a patient. Okay. One non-silent mutation in a gene in a patient. So I'm not sure that you would gain a lot by adding counts. But there may be, for cancer types where you have a very high mutation rate, then it may be the case that using counts could be a better, give you better results. But at least for the ones that I've looked at and the studies that I've looked at, usually have only one mutation in one gene in one patient.
More? No? Okay, so if I understand correctly, now you have the journal club, right? Then there's the lunch break, and we're supposed to be all back